Hi, I'm Dr. Deanna Hagge, and I work at an Andaman hospital in Kathmandu, Nepal. So, for instance, COVID will develop into new strains through mutations. Yes, the same can happen for bacteria. So, for instance, if there's changes in key genes that would be sensitive to a drug, mutations occur there, and the bacteria may become resistant to the antibiotics. So leprosy has been treated with 3 antibiotics for the last 40 years, and the first antibiotic for leprosy was discovered in the 1940s. So these are drugs that have been in use with leprosy for many decades. And for instance, dapsone, the first drug that was discovered for leprosy in the 1940s, by the early 80s, there are already reports of drug resistance to dapsone. And so in the 1980s, multi-drug therapy was issued or made available globally. So that combined dapsone with rifampicin and clofazimine. And by putting 3 drugs together, you decrease the risk of any bacteria that is treated with them of being resistant to all 3. So in leprosy, we've been using these 3 drugs, and MDT has been successful. But the strongest and most active drug in there is rifampicin. And there are rifampicin strains, or rifampicin resistant strains out there from TB, and there's also rifampicin resistant strains for leprosy that are out there. And then to back up the 3 drugs that are in MDT, there are second-line drugs. And so we are part of a program with the WHO that provides surveillance in different countries to test and see, particularly in relapse cases, if they're carrying drug-resistant leprosy in their body. And did that happen because it didn't respond to the medication the first time, and then it developed into a secondary infection, or what? So this occurs in a number of countries around the world, from Brazil, India, several sites, Nepal, and several countries in Africa. And samples are taken, and we report those every year or so. And the WHO issues a report as an indicator of what drug resistance is currently out there. And so rifampicin is one of the main ones that people worry about, because we know dapsone drug resistance is already out there. So dapsone and rifampicin together starts to cause a problem. That information provides an idea of how much drug resistance in leprosy is actually out there in the field. But we're actually taking very small samples in endemic populations. And for instance, some researchers in Delhi and in North India found that there were some ENL cases that were not responding to reaction treatment. So ENL cases are patients that generally have very high bacterial loads, and these patients would have already been treated with MDT, and so they're trying to treat them with other drugs to calm down the leprosy reaction episode, but they're not responding properly. And they found in North India that when they added another antibiotic, their leprosy reaction calmed down. And so further tests were done, and this was included with the Leprosy Mission Delhi lab, and correlated that some of these were drug-resistant strains. So you have patients that have high bacterial loads of leprosy. They're then receiving a year or more of MDT, but they're still having live leprosy in them. And then it takes a while for it to be determined that that's grown up. But the whole time, they're walking around with drug-resistant leprosy that may not respond to MDT in other people, and it starts a chain reaction and a spread. So noticing cases, multiple cases in North India with ENL, that was also not responsive to treatment until a different antibiotic was given is a problem. We will be meeting after ILC, the International Leprosy Congress, again, to come together as countries who do this surveillance and report our data, which then the WHO will put into a report that can be read globally. So it keeps a pulse. It's like a vital signs check that keeps a pulse on which types of drug resistance are in which countries, and are they increasing in frequency. So, for instance, oxyfloxacin is another antibiotic that can be used as a second-line antibiotic. However, there are issues when you start talking about which antibiotics are already in the population and being used for other diseases, and particularly in some populations where antibiotics are given freely or people can access them and take whenever they want or stop taking whenever they want, and those could impact how drug resistance is developing within particular populations. You want to protect drugs from being too much in the population and causing selection for drug resistance. For instance, if people in the population are taking a certain antibiotic for other sicknesses and there's leprosy in their system, that antibiotic may kill some of the bacteria that's susceptible, but it will selectively keep the ones that are resistant growing. And then that person may not know that they have leprosy, and so it will grow and potentially transmit to other people. So just like in some countries where they tell doctors not to give out too many antibiotics because you can reduce which antibiotics are going to be most effective for strep throat, for staph, and things like that, that is there similar for leprosy as well. So for instance, currently, we are primarily scanning relapse cases. So these are people who had leprosy, and then they were treated with the MDT, and then some years later, they become positive again for live leprosy. So the question in those patients is, did they have drug-resistant strains when they took their MDT originally, or did they pick up a second infection after the first cure? So if you're testing in those patients, those are the ones that are at highest likelihood to have drug resistance appear in them, right? Because they've previously been treated with antibiotics. However, one of the problems is that when those patients, relapse cases, have passed it into the general population, and you have people who've never been diagnosed with leprosy, and yet they have drug-resistant strains in them. And what that means is, because leprosy grows so slow, they will put that patient on a regular MDT, which will kill bacteria in there that are susceptible to the antibiotic, but the ones that are resistant will keep growing. And it may take a very long time to actually determine that that has happened. And as that's happening and growing, it's increasing risks for infection in nerves, nerve damage, which can lead to disability and deformity. So when we say that the best thing we can do for patients is find them and diagnose them early, a sideline on that is we need to be able to have the best antibiotics to treat patients properly as soon as we find them. Could there be a different MDT in future? Yes, if we're able to identify that there are better drugs that are more effective against leprosy and maybe shorter duration or have less side effects or just better antibiotics. Until then, MDT, as it is relatively effective, but we need to be watching drug resistance that's developing in populations.